Okay, so we are getting closer to our first exam. We're a week from today, our first exam. So I am planning on most of you taking that exam right here in person. If you're at home listening to this, I would prefer if you send me an email if you get quarantined or whatever. I'm going to make it available on Respondus as well. But I want you to communicate with me if you plan to take it that way. And obviously I want you to have a good reason. So most of you are going to be taking it here. I did post just a little Respondus practice. If you haven't used it before, or even if you have, just make sure everything's working right. If you are going to be taking it by Respondus, then you're going to need to take it exactly the same time as the rest of the class. So 10 o'clock next Tuesday. So be kind of ready to do that. A couple of people have what seems like a pretty good excuse, whether it's anxiety or whatever else, that they would like to take the exam in a smaller group setting. And I can do that for a few of you. So if you're interested in maybe taking it in a conference room or something with a few other students, I can arrange that too. So just come and talk to me. If you're nervous about taking it in the big class, I might be able to work with you a little bit in that way. Okay. I also did, or I will today, I have it all kind of ready to go. It's a 10-question just practice test. These are questions that I've used on exams in the past, and just so you kind of see what kind of questions that I ask. So I'll get that posted today. I haven't decided exactly where I'm going to post. I'll probably put it with the exam objectives. So look for it in the tab there. I'll change the name to exam objectives and practice questions for exams, because I do that for every exam. Okay. So keep working on your homework. Some of you are, some of you aren't. So don't wait till the last minute on that. Okay, so that was my announcements. Okay, question. Oh, how many questions will be on the exam? I have it pretty much ready to go, and I have 62 questions. So you have more than a minute per question. I think, like I said, you have plenty of time. I often give the same length when I teach a Monday, Wednesday, Friday class. So you should have a good amount of time to do that. A couple questions, or at least one question about, we talked last time, we finished up the structures of bacterial cells and eukaryotic cells last time, and in comparing the flagella of bacteria and eukaryotes, we talked about how there's tons of differences. Really the only thing the same is that flagella is for motility, and it helps move the organism around. So of course, you don't need to know anything more about these structures, but I said the bacterial protein flagellin that makes up a bacterial flagella. I just threw up the structure there just to kind of eyeball the difference between that protein that makes up eukaryotic flagella tubulin. You know, to me, looking at that, you know, that doesn't tell me a lot of information. Of course, totally different amino acid sequences, different shapes. The tubulin is usually associated in a dimer with another tubulin, and there's different variations of both those proteins depending on the organism and all sorts of things. So it does get quite complicated from here, but just had a question, so I thought that I would throw that up there. All right, we ended last time, we were just starting our survey of eukaryotes, and we were just talking about some different characteristics that a lot of the protozoa share. When we left off, I was saying that most protozoa are free living, meaning, well, free living mostly in bodies of water, sometimes in moist environments, but a few of them, okay, this is going to be a problem. Okay, just a second. Let me see if my pen should not be out. Okay, that's not it. Oh, I turned pen on. Okay. Sorry about that. So most of our protozoa are free living. Some of them are parasitic. And just a few examples. We're going to talk about one in a little bit more detail, but we've got the Giardia here. We talked about how that one causes diarrheal disease. The trypanosomes, those cause sleeping sickness. And the one that we're going to spend a little bit more time on here is malaria. So I like to talk a little bit about malaria. Actually, before I do that, I'm not going to make you learn a whole bunch of different life cycles and structures for protozoa, but there are two terms that seem important because all protozoa have at least one structure that we can describe that's called a trophozoite. And when I was looking through these slides last night, I thought this slide looks really bare, and so I threw in a picture here that I know you don't have on your posted slides, just to kind of visualize it a little bit. But a I can't even say it, trophozoite stage of protozoa, this is the motile feeding stage. Okay. So all protozoa have this, whether it's motile because it's an amoeba and it's, has, pseudopods that it extends, whether it has flagella, whether it has cilia, that's the stage where it's moving around and it's eating. Okay, this Giardia, this is the trophozoite stage here of the Giardia, one that it's moving around. Okay, some bacteria, or some, I'm used to saying that, some protozoa can exist as a cyst, and that's a hardy resting stage. Okay, so usually it's going to be probably not perfectly round like that, but some kind of roundish shape. It's not the same structure as an endospore. So remember, some bacteria can form an endospore. That's a super strong structure, just survival mechanism, can last for millennia. We don't even know how long. Cysts are not that hardy, but they are kind of a dormant stage, again, that some protozoa can kind of hunker down in. All right, so I want to talk a little bit more detail about malaria, mostly because it's just so interesting. Your book, I used to talk a lot more about fungi, and your book like dropped some of that information off, so I was just as glad to do that and replace it with an interesting protozoa. Why do I want to talk about malaria? Well, it's the single greatest travel threat for Americans that are traveling outside of the country. About half of the world's population lives in areas where malaria is endemic. And that means just kind of constantly being passed around. Okay, so malaria is caused by this protozoa, a species called Plasmodium. And of course, that is carried by mosquito, but we'll get to that in a minute. So there's 5 different species of that Plasmodium protozoa that can cause malaria. And I don't want you to memorize the genus and the species, or the genus you should recognize. You don't need to recognize all those species names. I mostly mention it because the severity of the disease that someone gets is going to depend a lot as to what species they're infected with. So the worst one here is the Plasmodium falciparum. That's the most severe form of the disease. And that is mostly what people get in sub-Saharan Africa. Okay, so that's the most deadly form. The second one here is the Plasmodium vivax. It's a little bit less severe. This is mostly Asia, Latin America, where that's mostly circulating. The malariae and the ovule, those two are not nearly as common. And this Nowlowski has only recently been realized that humans can be infected with that species. That's mostly been a species that primates get. And we've had a few instances where we've realized that that can cross over into humans. So that's, again, only a few cases. All right, so that Plasmodium is dependent on a particular species of mosquito here, Anopheles mosquitoes. So we're gonna look at the life cycle in a minute, spend a few minutes on that, but basically the Plasmodium develops in the mosquito passes it to humans, part of the life cycles in humans, and then we pass it back to mosquitoes. And it's female mosquitoes only. They're the only ones that take the blood meals that need that to feed their developing eggs. And the interesting thing is that the mosquitoes are not harmed in any way. If only they were. Okay, so we try to get rid of the mosquito in order to actually cut down on the protozoa. That's usually the strategy. What does malaria look like when humans get it? Again, it depends on what kind that you get, but usually a couple days after you're bit by a mosquito, you get this disease that's cyclic. So we go through these 3 disease manifestations, go through all 3 of them in about 6 to 10 hours, and then 2 or 3 days later, you go through the cycle again. So it starts out with a cold stage, so that means the patient is shivering, you got the shakes probably, and that then goes into what's called the hot stage, very high fever, often have vomiting accompanying that, really bad splitting headache. And then the fever breaks in the sweating stage, and patient is just exhausted. Okay, so it's cyclic. Now that doesn't really, it sounds kind of uncomfortable, right? It doesn't really sound that bad. I went on to the Gates Foundation. Bill Gates and his wife have done a lot of work in trying to alleviate the effects of malaria. And here's an account of someone that had malaria and how he felt. I awoke to what felt like lightning going through my legs and then spreading through my body and head. Probably the worst headache, body aches, and chills you could possibly imagine. It felt like I was being stung repeatedly by an electric shotgun and could barely control my movements. The pain was so intense, I actually believed I was dying. Okay, so not fun. There's lots of other complications that can happen, kidney damage, anemia, because this is replicating in our red blood cells. There can be neurological impacts. I had a friend that had malaria a few years ago, and he was in a coma for a long time and came pretty close to not making it. So mostly this is the mortality is definitely worse in kids under 5. So malaria kills about somewhere on the order of 429,000 people in the world every year. That's figures according to the WHO. Over 200 million cases though. So lots of problems. In the US right now, we don't have malaria passing between people. We have travelers coming in, maybe a couple thousand a year. We used to have malaria in this country, but we are able to eradicate those Anopheles mosquitoes, mostly with the help of DDT. So, of course, we don't use DDT anymore, and there's a big controversy over whether to bring that back on a small scale, spray it inside homes, that sort of thing, and just not spray it widespread in the environment. Because in many countries, malaria's really come way back due to those mosquitoes making a comeback. Okay, there's a huge worry this year that because of COVID, that's going to disrupt a lot of the programs that we have in place to keep malaria down. like passing out insecticide sprayed mosquito nets and making drugs available to people. Those programs aren't happening because of COVID, so we're really worried that malaria is going to really have a huge uptick this year. All right, looking briefly at the life cycle, and I don't want you to memorize this. I want to point out some highlights here. Of course, malaria has been a disease that we've known about like as long as humans have been around, an ancient disease. It was only within about the last around 1900 that we figured out the life cycle. Extremely complex life cycle. So I want to point out a few things that are interesting. If we start right here where the mosquito takes a blood meal. So we have a mosquito that's infected with the Plasmodium, and it bites a human host. Okay, so when mosquitoes bite, they have actually like multiple proboscis that they actually bite you with. One tube sucks out your blood, another one injects their saliva, and in the saliva you've got the Plasmodium as well as the anticoagulants, so they can actually suck your blood out. So they inject the Plasmodium initially. Right here, that protozoa is going to infect the liver. Okay, so part of its life cycle is in the liver. It says exoerythrocyte. That means outside the red blood cell cycle. Okay, don't worry about the terminology here. I'm not too concerned about that. Eventually, that part of the life cycle matures, it ruptures, and then it enters humans' red blood cells. Okay, and that's when you start to see all the disease manifestations, when we've got that part of the life cycle in the red blood cells. Okay, and here you see that trophozoite, okay, that motile feeding stage, that's inside our red blood cells. All right, and so it actually produces gametes. I'm not too concerned about that, but over here then, when another mosquito takes a blood meal from somebody that's infected, then those gametes develop in this over here is in the mosquito. Here we have a stage where it hangs out for a while, and a cyst stage. Okay, so that's kind of the highlights there, initially in the liver and then enters into the red blood cells. Okay. Questions? Okay, another interesting thing that I got from that Gates website, and this is just because it's interesting. If you think, and I didn't post this, but if you think about the world's deadliest animals, what comes to your mind? Probably like a lion or a shark maybe. Well, shark, kind of at the low end there, only 10. So all your big mammals, we've got some parasitic infections, multicellular parasitic worms in there. Freshwater snail, this actually carries a worm, a little parasitic helminth worm that causes schistosomiasis, but that worm couldn't be there without the snail, so I guess they count the snail. You've got dog bites, rabies, lots of developing countries, that's a huge problem. And venomous snakes, 50,000 people. Humans killing lots of other humans, but by far the biggest killer there is going to be the mosquito. There's a really great book over at the library, good book, we'll say good book, over at the library called The Mosquito. And it talks about, there's like 14 different infections that mosquitoes carry. So malaria is a big one, of course, that's protozoa, lots and lots of other viral infections, yellow fever, dengue, Zika, West Nile, Eastern equine encephalitis. Remember a year ago, everybody's all worried about EEE, changed football game times, ooh, and now it seems like not anything, but people died in West Michigan because of that last year. I saw an article a few weeks ago that they found some horses infected. So anyway, all sorts of things, not just malaria, that the mosquito carries that's deadly. So just sort of interesting to think about there as an aside. All right, so any questions about Protozoa? That's all I got for you on Protozoa. We could probably go further on that and it would be interesting, but I just want to spend a few minutes here on algae. We're not really as interested, in this course, we're not as interested in algae because, of course, it's not ever harmful to humans, with exception of red tide, right? So if you get an overgrowth of nutrients, sometimes it's after a storm that kind of wells up the nutrients from the seabed, you get an overgrowth of algae, and then enough toxins are produced that can be harmful to marine life and to humans as well. Other than that, right, algae, we get a lot of oxygen from photosynthesis of algae, especially in our oceans, you know, forms a basis of lots of food chains, marine food chains. So, you know, it's a great thing and useful thing to humans, but we won't, again, not typically disease causing. Of course, so eukaryotes are photosynthetic. They can be unicellular, like the diatoms here. They can be colonial, like Volvox. You might have looked at that in biology at some point. Algae can be multicellular. So here's some red algae. Remember, that's what we harvest agar from, the cell walls of red algae. This giant kelp here can stretch for miles across the ocean. So it's not just that algae are little and plants are big. The difference between plants and algae, and this is more like just if you're interested, it's the reproductive structures. In algae, I think every cell in algae can be a reproductive structure, but it has potential to be a gamete. But in plants, it's only certain parts of the plants that can be reproductive. So if you're interested, that's the difference there. Okay. All right, so we're going to spend a little bit more time. Oh, wow. One other interesting thing I wanted to share with you. What about plankton? That's a whole like taxa that we haven't mentioned. What is plankton? Is it a bacteria? Is it an algae? Is it a protozoa? Well, actually, it's all those things. Plankton is defined by where we find the organism. It's plankton are microscopic organisms found in a large body of water. So it could be algae, it could be protozoa, bacteria, archaea, any of those things are all plankton. So in case you were wondering about that. All right, we're gonna spend a little more time on fungi. There are lots of parasitic fungi. We're gonna spend some time looking at structures, a lot of new terminology with this little time that we're gonna spend on fungi here. We're gonna go into their reproduction a little bit because that's how they're classified. So if we think about what makes a fungi, we've maybe mentioned this before, their cell walls are primarily composed of chitin. Okay, that's a polysaccharide that we often find in their cell walls. It's not a hard and fast rule, I don't think, but that's very common. And of course, they do not photosynthesize. Okay, so that's the big difference between plants and fungi. Why do we want to study fungi? What's the significance of this? Can you think of a way that humans use fungi? Throw anything out there. Make bread, good. Food, right? Personally, I don't like mushrooms on my pizza, but a lot of people do. Nutrition source. I heard somebody else over here. Drink as well, right? Saccharomyces cerevisiae, alcoholic beverages. I'm surprised some of you are probably thinking that. What else? Drugs. They are a source. There are fungi, Penicillium, the genus Penicillium, that synthesizes antibiotics. So probably at least half of our antibiotics are made by fungi. Let's see, they're decomposers. So responsible for nutrient recycling in our environment. We'd have a lot of dead stuff around if we didn't have fungi to decompose those. One that you might not have thought about, there are lots of fungi in symbiotic relationships with plants, especially. The organism that we know as a lichen is actually an algae and a fungi that live symbiotically together. Okay, so that's one example. I'm going to introduce, there's a term here, mycorrhizae. Whoops, 2 R's, 1 H, sorry. Okay. That's a term I want to introduce. That's a symbiotic relationship between the roots of plants and fungi. And this is, we think, extremely common. We think that most plants actually grow with the help of fungi, making these mycorrhizae with the roots of plants. So it helps, fungi actually help plant roots absorb more water. So here's an example up here. I pulled this off the web somewhere, of some grass that has those mycorrhizal fungi and grass that doesn't. Okay, so it's kind of hard to tell, but the root system here is a lot more robust, and ideally, and then, you know, the grass is healthier because it has these good strong roots. Okay, something bad that you could think of course, that fungi do also cause disease. So things that are fairly not too much trouble, like something like athlete's foot. We have plant diseases as well, like Dutch elm disease that's taken out all of our elm trees. But there's some very serious fungal infections, although they're not really very common, like coccidiomycosis. It's a pneumonia. It's difficult for us to get rid of fungal infections because, of course, they're eukaryotic cells, and we just don't have as many options of drugs that are going to target the fungal eukaryotic cells and not harm our own eukaryotic cells. All right, so let's talk a little bit about some fungal structures. Again, apologize, lots and lots of new terminology here. Probably, I'm assuming you probably haven't looked at fungi too carefully in other classes. Our fungi, let me just write that up here. Let's see. When we're thinking about fungi, the three types, we could think about mushrooms, molds, and yeast. Whoops, yeasts. Okay, so that's kind of the three types of fungi. If we look at structures that are molds or fleshy fungi, I guess we refer to mushrooms as kind of a fleshy fungi. What kind of structures do they have first? Well, they have hyphae. So basically the cells grow in filaments. Filaments of cells. Okay, and those hyphae are either going to be septate or aseptate. Okay, septate hyphae, that means that they have cross walls, and aseptate hyphae have no cross walls. Okay, that A means not or without. All right, so this picture is not great here, but the one on the left here, the septate hyphae, you can kind of see, not exactly sure where those hyphae are supposed, or sorry, those septa are supposed to be. You can kind of see there's some distinct cells there, where the one on the right here don't have those cross walls. So basically, if it has a septate hyphae, there are pores there that allow nutrients to be transferred between the cells. But when there's no cross walls, everything just kind of flows in between those hyphae. So because there's no cross walls here for the aseptate hyphae, basically it's considered multinucleate. Okay, so you have all the nuclei are just kind of flowing around through there. We have another term for multinucleate, and that's cenocytic. Make sure I spelled that right. Yep. Okay, so cenocytic, that means multinucleate. So if you have aseptate hyphae, that's also considered a cenocytic hyphae. Okay, so lots of terms. Hyphae, again, they're either one or the other here. Hyphae can also be vegetative or aerial. Okay, so again, one or the other. Vegetative hyphae, their job is to obtain nutrients. And we'll talk about how they do that shortly. They form masses of hyphae, another term. They all grow together. Called mycelium. So let's see. There's another new term, cenocytic there. Okay, so I'll show you a picture of a fungal mass, massive hyphae, mycelium in just a second there. Aerial hyphae, these are going to be the reproductive parts. Okay. And that's going to be the source of aerial spores. Okay, so sporulation in fungi, that's how they reproduce, at least the filamentous ones. All right, so here's a picture from your book of just a fungal mycelium. All right, so masses of hyphae, those hyphae branch out and fuse with other hyphae to make these huge masses. If it's a mold, then you would see this part. Mushrooms make mycelium as well, but it's usually going to be underground. So you have these, big underground network of mycelium. That's why you might overnight notice a mushroom suddenly appear in your yard. Well, the mycelium was underground, you just couldn't see it, stored lots of nutrients, and then when the conditions were right, bam, that mushroom grew really quickly. So that's where the term mushrooming comes from. Okay. I found this little graphic on the web. Let's see. Yeah, I did post the site there. Just because I liked it. It went through some of that terminology again and let you see it in a little different way. Spores, remember this is the reproductive part. The spores, when they germinate, so they grow hyphae typically out of the spore. Here's mycelium, remember a big mass of hyphae. We're going to talk about this structure in just a second, Hostoria. Cenocytic hyphae then are aseptate, no cross walls, septate hyphae. These, I believe, like this structure right here, that's supposed to be an aerial spore growing off of a hyphae. And here's another method of reproduction that yeast do. It's called budding. We're going to talk about that next, okay? And this is just one of the kinds of spores. We're going to talk about that again in a few minutes, so we'll come back to that. And I would say don't really worry about those terms down there. Okay, so we talked about filamentous fungi. Yeast have a little bit of their own terminology. Of course, yeasts are small and globular. They're usually not perfectly round. If you see yeasts under a microscope, they're going to be quite a bit bigger than, say, bacterial cocci, usually not perfectly round. And they usually reproduce by budding. So that means you get kind of a, when bacteria divide, they basically split in half. We're going to talk about that when we get to growth pretty soon. Usually just like a little bit of the cell will kind of pinch off when we get budding taking place in yeast. Okay, if the bud fails to detach, then that's called a pseudo-hyphae. Okay, so that's what this picture is showing here. Some buds that are still attached. We call those pseudo-hyphae. Okay. We think that that can help organisms penetrate through cracks. Okay. Some organisms can produce both the filamentous and the yeast structure, and we call that dimorphic growth. Okay, so that's yeast-like and mold-like or filamentous growth. Often, so here's a picture here of an organism that's making both yeast-like growth and filamentous growth. Typically, the organism wouldn't be making both of those at the same time. So it might be switching over from one form to another. Which form it makes is usually triggered by something in the environment. Often it's going to be temperature dependent as to which form it takes. An example of an organism that does dimorphic growth, Candida. So that's a very common fungus. It's the one that usually causes athlete's foot. It often causes thrush, which is overgrowth of fungus on the tongue. It's not as simple as, well, when it's in the body, it forms this form, and when it's in the environment, it forms this form. I think usually, again, this is not a hard and fast rule, that if the fungi is in our body, it's in a yeast-like form, and if it's in the environment, it's a filament. But I don't think that's a hard and fast rule, but I think that's commonly the case. Okay, how are fungi getting their nutrients? Okay, I mentioned this term before. We're going to go into a little more detail this when we get into chapter 6. But most fungi are chemoheterotrophic, and that means that they get their nutrients through organic sources. Okay, they eat stuff. That's just like what we do. We're going to pick apart that term a little bit later. So a lot of fungi, most of them, we'll say most, are considered saprophytic. Whoops, there's a T in there. Saprophytic. Okay, so that means that they absorb their nutrients from dead sources. So we talked about how fungi, lots of them are nutrient recyclers. So they decompose whatever, logs, things like that are dead. We used to have some shelf fungus that would grow every year on a tree in our front yard. It's usually a bad sign, because it's only going to grow there if there's something dead in the tree there. Some fungi, however, are parasitic. And that means they get their nutrients from living organisms. So these are both kind of a form of being chemoheterotrophic, you know, how they get their nutrients, whether it's through dead things or alive things. If a fungi is parasitic, it typically will have modified hyphae called Haustoria. Okay, so in this picture here, you can kind of see this down here. Basically, if a fungus is going to get its nutrients from a live organism, it has to be a lot better at penetrating into that live organisms. Intact cell walls, intact cell membranes are a lot tougher to penetrate than an organism that's dead. So they typically have these, you know, a little bit, whatever, stronger hyphae called Haustoria. And if it does, if the fungi is parasitic, then it's going to cause some kind of fungal disease that's usually referred to as mycoses. I've got the C in there. So that's a fungal disease. All right, so spend just a few minutes here on fungal reproduction. Again, this is important because this is typically the way we classify fungi based on what kind of reproductive structures that they make. So the good news is I used to do a lot more of these and I pared it down a little bit, but a few different kinds of spores. I want you to be able to recognize those three and what they look like. So all fungi do all fungi make asexual spores. And they're making like hundreds of spores, basically. So remember, unlike bacteria that would only make one spore, not reproductive in bacteria. But all fungi do this. Of course, the progeny are going to be genetically the same as the parent. So if the conditions that the fungi is in are ideal, they're just going to keep making more and more of themselves that's exactly the same. So don't mess with success. All right. So three major forms of asexual spore formation. The sporangiospores, that's the first one pictured up here. The spores are going to be located in a sac. Or enclosed in a sac. Okay, so when the time is right and those spores mature, that sac bursts open and releases those spores into the environment, and they germinate into typically hyphae. All right, chlamydiospores is a little bit different because that spore structure develops within the hyphae. Okay, so it's right here, chlamydiospore within that hyphae, and again, when that matures, that will burst open and release lots of spores. And then what's different about the conidiospore, and this is probably the most common spore structure, is that the spores are not enclosed in a sac. Okay, that prefix conidio apparently means dust. So basically, any kind of movement in the wind is going to kind of disrupt those spores, and they'll get dispersed in the wind, I guess. Okay, so I think the sporangiospores and the conidiospores are more common than the chlamydiospores, but I think the conidiospores are then the most common. Okay, so I want to lastly just go through sexual spore formation a little bit, talk about that a little bit. If conditions are not quite ideal, then some fungi can reproduce sexually. Of course, the progeny are going to be a little bit different than the parents, so they may be better suited to adapt to some environment that's maybe harsh in some way. So again, if conditions, let's say if conditions are not ideal, then some organisms can sexually make spores. So going through kind of the general cycle, how that would happen, how that sexual spores would get formed. Let's start up right here, and I tried to leave my narration here a little bit. I see my numbers got kind of disappeared here a little bit. 4 points there that I left in. Let's start up here. So you have two hyphae that fuse. And we don't call hyphae male and female. We call them just, if they fuse, they're compatible. We say a plus hyphae is compatible with a negative hyphae. Okay, so not male and female, but compatible tips fuse. We can't tell by looking at them if they're compatible or not. When those tips fuse, then this cell right here has a nuclei from the positive and a nuclei from the negative. Okay, and we call that a dikaryon. If you do a karyotype, that's where you can like see the chromosomes. So that term karyon has something to do with genes. Two nuclei there. Okay, so we call this this part in the orange here, we call this the dikaryotic stage, and it's also known as the N plus N. So the plus chromosomes and the minus chromosomes, again, they're staying separate in that one dikaryon. That dikaryotic cell reproduces itself by mitosis for a while, 2 nuclei in each cell, but at some point those 2 nuclei fuse, and now we call the cell diploid, okay, 2N, 2 copies of the chromosomes. So forget your basic algebra, N plus N is not the same thing as 2N. Okay, I know that's a little bit confusing. Okay, so now we have diploid nuclei, 2 copies of each chromosome, and at some point that starts reproducing, going through mitosis. So remember, mitosis halves the number of chromosomes, so you get half the number of chromosomes in each of the spores that are produced there. It's producing both plus and minus spores. Those spores germinate into hyphae, and then the tips fuse, and we start all over again. So this is the haploid stage over here. Okay, so we could go through the different kinds of sexual spores that we see, or structures that we see, but we're not going to worry about that. You have a nice video clip, I think it's part of your homework, that goes through this cycle again. So again, you get another look at that terminology as part of your homework. It's one of those Dr. Bauman's video clips that are really very good. So take the time to look at that and reinforce that terminology. Okay, so I know there's a lot of new terminology again with that fungal stuff, but just review that. You will see some of that again on the test, so don't skip over that. Okay, I wanted to take just a minute and give you a couple of review questions. I probably wouldn't put a question exactly like this on the exam because it would take up a lot of space on a printed exam, but you're going to see some questions that are just really straightforward. What is this? What is that? That's supposed to be sort of your lower level of recall type questions. Which of the pictures represents the Lophotrichus flagellar arrangement? Excuse me. Okay, so this one up here is the Paratrichus. Remember, all around. We could call this monotrichus or polar flagella. It's just at one end. Here we've got our Lophotrichus. So remember, that means the tufts of flagella. And this one is Amphitrichus, ampha meaning at both ends. So just a little bit a straightforward question. And one other question before we move on. Which of the following statements does not support the endosymbiotic theory? I actually had this question on an exam and then I decided to throw it out at the last minute. But you're going to have some questions that require a little bit more complex thinking and definitely will throw in some detail from what we've done as well. So I'll give you a second to read over that and think about which one you think it is. All right, so if we go through this, let's see. Mitochondria have circular DNA, yes. But we're looking for what does not support the endosymbiotic theory. Let's see, most mitochondrial proteins are encoded by nuclear DNA. That is actually true. And I said that, but I'm not sure if I wrote that. That's why I ended up throwing that question off. But that's true, and that actually doesn't support the endosymbiotic theory. Remember what that theory basically tries to explain how a eukaryotic cell emerged from two prokaryotic ones. Okay, chloroplasts have that bacterial ribosomes. Yes, that's true. Yes, all these things are true, but again, this is the one that doesn't support, so. Okay. All right, questions before we move on. Okay, so we did a little bit of chapter 6 before. We're going to hit the rest of it now, microbial nutrition and growth. We're going to start out, and I did, let's see, I posted this over the weekend. I know it looks like we're a little bit behind from the syllabus. I think we will have to bleed into next lecture to finish up chapter 6, but hopefully that's okay. I am planning on the exam covering through this chapter 6 material. Again, we'll start it today and finish it beginning next time. Okay, so we're looking at growth requirements for cells. So that's important if we're trying to grow organisms up in the lab, that we know what kind of nutrients to give them. If we're trying to kill organisms, maybe what do we want to deprive them of? We'll look at different environmental factors that are going to influence growth. Some of that's a little bit intuitive, I hope, but it does help you kind of learn to think like a microbiologist. What kind of parameters do we need to provide to grow organisms? This is the only time we'll do math, right? So we're going to look at some numbers in order to help us kind of understand how cells grow. And along with that, graphing out growth rates. So just kind of the basics there. And of course, we need to have some way to count cells and understand the different ways that we can do that. That's all going to be part of it there. So, okay, so what kind of nutrients do organisms need to grow? Same kind of nutrients, of course, that we need is what bacteria needs. Macronutrients, these are the nutrients that organisms need in large quantities. Okay, so carbon, hydrogen, oxygen, that's sort of obvious. Basically, we'll just say for cell structures. Okay. Okay, so we need large amounts of those elements. Nitrogen, if you think about what in the cell has a lot of nitrogen in it, for proteins, of course, amino acids have that amino group, right? So lots of nitrogen in proteins, nucleic acids. Okay, nitrogenous bases in nucleic acids, so lots of nitrogen in those structures. Phosphates, there's lots of phosphate in DNA. Membrane phospholipids. Okay, also ATP, adenosine triphosphate. Okay, and then potassium, magnesium, calcium. Iron sometimes is considered a macronutrient, sometimes it's considered a micronutrient, kind of depends on who you ask there. So I put that one in parentheses, but these are all elements that are necessary for enzyme function. And apparently cells need a good bit of them. Okay, there are nutrients that are needed in much smaller quantities. They're no less important, the cell just needs less of them. Yes, question? What's that? Yes, I can go back for a second there, yes. Yeah. All right, so cell needs them, no less important, just doesn't need as much of them. So all of these micronutrients, and sometimes they're called trace elements, they're all metals and mostly necessary for enzyme function. Okay. And then what kind of growth factors a cell needs, it really depends a lot on the cell. Lots of bacteria, or at least some bacteria cells, can manufacture a good bit of the organic things that they need. So certain cells can manufacture some things and other cells have to get those things from their environment. So amino acids, coenzymes like NADH, we're going to talk more about that molecule next time. It's an electron carrier, certain vitamins. So those are all things that might be necessary and be considered growth factors. All right, so I said that we would revisit that term, chemoheterotrophs. You think about how do organisms get those nutrients that they need. These terms are inherently confusing because we have two prefixes that we amalgamate together to make a word that's supposed to be meaningful. So the root here, troph, that means nutrition. Or sometimes you could say that root means to feed or nutrition. Okay, so that's the root there. And then how do cells get their nutrition? We could either describe it, well, or we'll describe it as how they get their carbon source. And we use the prefixes auto if it gets the carbon source from carbon dioxide, and hetero, which means other, if they get their carbon source from organic compounds. And then we also try to understand where organisms get their energy. Do they get it from the sun, from light? Then we use the prefix photo. And if we get it from some chemical compound, like the breaking of chemical bonds, we use that prefix chemo. Sorry, allergy day. Okay. All right, so I talked about how most organisms are going to be considered chemoheterotrophs. Okay, that's the most popular method. Most organisms, most bacteria even, are going to, they're basically going to eat stuff. Okay, so here's some examples of organisms that are chemoheterotrophs. Most protozoa, most fungi, of course, we are. The other popular method is this photoautotrophs. So all of our green plants that photosynthesize, there's lots of bacteria that do that as well. Even some protozoa are photoautotrophs. And then these last two categories are mostly composed of really oddball bacteria that have really unique kind of metabolism. Okay, so I don't think, they give you some examples here of organisms, these chemoheterotrophs. Some of those organisms live like at the bottom of deep sea vents, bottom of the ocean and deep sea vents. So just some kind of weird environmental organisms, some archaea that do that. So I won't worry too much about examples there. Okay, and we could throw in another prefix there based on how organisms get their electrons, but I decided we're not going to worry too much about that. Okay, so if we start looking at environmental influences of growth, so how do we get some bacteria in the lab, say, to grow faster, or how do we slow down the growth of bacteria? The very first thing we always think of, because maybe it's the easiest thing to manipulate sometimes, is the temperature. Okay, so most bacteria have a range of temperatures that they can grow at. For bacteria that would live in our bodies and cause disease, their ideal temperature is going to be near body temperature. And then typically a little bit higher than that, the growth really tanks out. Okay, that's because this is where the enzymes are working most efficiently, and usually after a certain point, those enzymes are going to start to denature. So if we want to slow down growth, obviously we would typically lower the temperature. And here's an example. I didn't post this. This is from your book. Here's some E. coli that's been incubated at 3 different temperatures for about 18 hours. You can see here at room temperature, it grows, but really slowly. Up a little bit higher at 30, we have a little bit better growth. I think it looks like it's a little bit better at body temperature here though. So again, an example of how E. coli is growing faster depending on the temperature, or slower. Okay, and we've got terminology. You go over this in lab as well when we test this in lab. But if you've got an organism that grows best at cold temperatures, and the temperature range, I wouldn't really worry about that, because I see that defined differently depending on different sources. But basically cold loving organisms we would refer to as psychrophiles. So room temperature and below. These are organisms that would grow well in your refrigerator, that would grow well on the polar ice caps, places like that. Mesophiles, all the organisms we use in lab are mesophiles. They're growing best between room temperature and body temperature. All of our disease-causing organisms are going to be mesophiles.